CRISPR was first observed and identified in E. coli. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. As you can see, there are two main parts found in CRISPR: the repeats and spacer DNA. The repeats are the short segment of DNA with 20 to 40 nucleotides that are repeating, interspaced and palindromic, meaning that its nucleotides are the same even when read backwards. On the other hand, spacer DNA are the gaps found in between the repeats. Different from the repeats, the spacer DNA are not identical from each other. Cas genes or CRISPR-associated genes are the ones that create Cas proteins that cuts DNA, but we'll talk about that later. CRISPR is a natural occurring method used by bacteria to protect infection by viruses. Here, you see a wandering bacteriophage looking for a host cell. When the bacteriophage attaches itself in the cell membrane, it produces its viral DNA inside the cell. The cell will then produce two types of short RNA, the CRISPR RNA and the tracer RNA. When combined, these two will form a complex with Cas9 protein. To form Cas9, use the tracer RNA to hold the CRISPR RNA in place, while the CRISPR RNA holds a segment of sequence that can be found in the viral DNA. The Cas9 will bind to the DNA and block a short sequence known as PAM. PAM stands for Product Spacer Adjacent Motif, a 2-to-6 base pair DNA sequence immediately following the DNA sequence targeted by the Cas9. Upon blockage, the DNA will be entwined by the helicase and caught by the nucleus. Mainly, there are two cases in which CRISPR-Cas9 can be used in genome editing. The first case is where the normal process happens. The cut DNA will naturally try to repair the DNA break, where it will include DNA repair pathways. This repair process involves insertion and deletion of nucleotides, which will lead to mutation. The mutated gene allows scientists to understand its function. The second case is essentially like the first, but instead of the natural repair process of the DNA break, a new RNA sequence called host RNA is introduced and attached to the DNA break, and make the break DNA complement the host RNA when repaired. Repaired DNA is called host DNA, which can be expressed and observed. The discovery and application of CRISPR-Cas9 is a huge breakthrough in the field of biotechnology. With the ability to cut and insert new RNA sequences, different gene-related diseases can be cured with little to none millstone in our body. That is all. Thank you for watching.